Now dear students, let us today discuss about identification of transformed cells. In the last lecture we discussed about how transformation is brought about. Once you have successfully carried out transformation procedure, now three different types of cells are formed. Okay, here the cloning organism is having the plasmid with insert, that means it contains the foreign gene inserted inside it, which is called as plasmid with insert. And in such cells, the cells are ampicillin resistant and the cells have non-functional tetracycline gene. How we will discuss. Now another type of cells are those which contain the plasmid but without insert. Such cells are not desirable because the foreign gene is not present inside the plasmid, so they are not desirable to us. The third type of cells are those which have not undergone transformation and which do not contain the plasmid, and such cells are not desirable. So the cells of the first type, that is cells having plasmid with insert, plasmid with foreign gene inserted inside, are only desirable. So what we need to do is, out of the three, all the three different types of cells are present in the cell culture of E. coli, but we have to identify only the first type of cells because they are transformed cells containing plasmid with foreign gene inserted inside it, and hence we need to study the identification of transformed cells. So let us discuss now. The identification of transformed cells is necessary because at the end of the experiment there are three possibilities. The host cell or cloning organism is successfully transformed by recombinant DNA vector with insert. These cells we want. The second type of cells where the cloning organism has undergone transformation but recombinant vector is absent. The vector is without any insert. So such cells are not wanted, not desirable. The third type of cells are the cells are not transformed at all. Hence identification of transformed cell is done because we want the cells of only first type, like this. Okay, now how to do? The vector used here for recombinant DNA technology experiment has two selectable markers. Say for example, we have used the vector PBR322. This vector has two selectable marker genes. One is tetracycline resistance gene and another one is ampicillin resistance gene. That means the cell having this plasmid will be resistant to both the antibiotics tetracycline and ampicillin. Okay, now these two types of resistance gene is active, right, and used for elimination of non-transformed cells. So the recombinant vector and E. coli cell suspension. E. coli cells are first treated with calcium chloride so that they become permeable, and the E. coli treated with calcium chloride are mixed with the recombinant vector. Now what is a recombinant vector? It is the plasmid PBR322 having foreign gene inserted inside it. Okay, and the cells are allowed to undergo transformation. The transformed cells are first cloned on, actually they are first grown on ampicillin containing medium to get a master plate. Okay, so got it? What we do is, we have an E. coli culture treated with calcium chloride so that the cells are permeable. In the second tube, we have recombinant plasmid, that is PBR322 plasmid contain foreign gene inserted inside it. Say for example, insulin gene inserted inside it. Now this recombinant plasmid are mixed with treated E. coli cells and the cells are allowed to undergo transformation. Once transformation process is over, we plate the cells. We do the plating of cells so that the cells grow and we can identify the transformed cell. For that purpose, first we grow the cell in ampicillin containing medium. Why? Because ampicillin resistance gene is present in the vector. Any of the bacterial cell having the vector will grow on ampicillin containing medium. If your cell is not transformed, it does not contain any plasmid, then the cells will not grow. So to eliminate non-transformed cells, we first grow the cell on ampicillin containing medium. Okay, and that plate is called as a master plate. To select the transformed cell having recombinant vector, the clones on master plate are replica plated on tetracycline containing medium. Okay, to select the transformed cells having recombinant vector, the clones on master plate are replica plated on tetracycline containing medium. Okay, and then once you have plated it on tetracycline containing medium, then we go for the next one, that is identification of transformed cell. Now when we are carrying out this experiment, what we do is, The foreign gene to be cloned is inserted inside the tetracycline resistance gene. As you can see from this diagram, we have a foreign gene. Okay, the foreign gene is treated with restriction enzyme. Okay, this is the foreign gene. It is treated with restriction enzyme so that you get fragments of the foreign gene. Then the fragments of the foreign gene are mixed with the vector, that is PBR 322 vector. Right. Now once you have mixed the foreign gene with the PBR 322 vector, the foreign gene gets inserted inside the tetracycline resistance gene. Why? Because we have used a restriction enzyme which will cleave the tetracycline resistance gene. So the foreign DNA gets inserted inside the tetracycline resistance gene and tetracycline gene is disrupted. That means now the recombinant plasmid loses its tetracycline resistance. But ampicillin resistance gene is intact. Now this plasmid is called as a recombinant plasmid. This is a recombinant plasmid. The recombinant plasmid is then mixed with E. coli cells that are treated with. So this is a recombinant plasmid. Okay, this is a plasmid having ampicillin resistance gene intact and tetracycline resistance gene broken down and the foreign gene inserted inside it. It is now mixed with E. coli cell treated with calcium chloride and plating is done on ampicillin containing medium. Okay, so what we do is plating of the medium is done on ampicillin containing medium. So this is a medium which contains ampicillin inside it, and all the cells, all the E. coli cells that have undergone transformation will grow on ampicillin containing medium. Okay, this is the medium which contains ampicillin. This medium contains antibiotic ampicillin. Okay, so non-transformed cells, okay, non-transformed cells that do not contain ampicillin, the non-transformed cell which do not contain the vector will not grow, only transformed cells will grow. This plate is called as master plate. Now, the colonies on the master plate are replica plated on tetracycline containing medium. Okay, the colonies on master plate are replica plated on tetracycline containing medium. Now, this is the master plate. Okay, the master plate is having colonies. The colonies of the master plate are replica plated on tetracycline containing medium. And now what you have to check is the colonies which are present on master plate as shown by the red colored box, but which are absent on replica plate are the colonies formed by desirable clones, and these colonies are selected for further experimental work. Now why? In our experiment what we studied that the foreign gene was inserted inside tetracycline resistance gene. That means tetracycline resistance was disrupted. So the plasmid having insert with foreign gene inserted inside it was the one having ampicillin resistance but no tetracycline resistance. Right? So the organisms or the colonies which are present on master plate but absent on replica plate are those where tetracycline resistance is disrupted, and they are formed by the desirable clones, cells containing plasmid with insert, and hence these transformed cells are desirable. They are selected out from the master plate, they are plated, and then further studies of expression of the foreign gene and production of protein product is studied. This is how we identify the transformed cells. The clones containing recombinant vector will be sensitive to tetracycline due to inactivation of tetracycline resistance gene by insertion of foreign DNA fragment. So the foreign DNA was inserted inside tetracycline resistance gene, and hence the organisms were resistant to tetracycline. That is why we select the colonies from master plate which are absent on replica